All right, you guys, I'm going to take my time so we can wait for everybody to come in before we actually start looking at the microbes. My name is Keisha Ernst. I'm a microbe farmer. I suspect that I'm in good company here. Walking around, I see every single table is talking about living soil, talking about microbes, talking about testing, and how we can optimize our growing systems to benefit microorganisms instead of killing them. Yes. Woo-hoo. So we've got 2 hours today to talk to you guys, and we've put a lot of effort into trying to give you some actionable ideas that you can take home and work with. The first part of this talk is going to be based on microbes. I want to take you on a journey through the microscope to see how it is that we look at samples, how we look at microbial communities and make sense of it. I want to point out this art. I'm blessed to be able to use it. This is Caitlin Solbeck. Her name's Protozoa Princess online, and she does fantasy art based off of what she sees through her microscope. And so, for me, so much of this is data, right? I mean, we've heard about SAP analysis today. We're going to hear about chemistry analysis later. Casey and I are both, we make compost. We are also soil food web consultants. So we help large-scale farmers, also at-home growers, landowners in general, learn how to alter their practices to be able to work with the microbes instead of against them. And so much of the work that we do is very data-driven. We're using chemistry analysis, we're using SAP analysis, we're using biological analysis. But my favorite part of the work is the adventure. It's looking through the microscope at all the different ecosystems around us and trying to make sense of it. And I guess it's a great way to start off is just letting you know, I'm going to talk about microbes a lot, and it's going to seem like I'm an expert, like I know so much. But you should all know, relatively speaking, I know nothing. I'm basically a microbial cheerleader that spends most of her life looking through a scope trying to figure it out. I believe 10 years from now, the understandings that we have about microorganisms and how they operate in the soil are going to be far more defined. They're going to be explained much better, and we're going to be in a place where we have a lot more information. So for now, the way that I look at soil is almost the way that I look at communities, the way that I look out into the audience today, and I see a lot of different people. I see people wearing different clothes, coming from different places. I suspect you all have different favorite foods, different ways that you contribute to the world. And I assume that all of you are out there doing your part to make this event an amazing event, right? We have the people that are organizing, we have the people cooking, we have the people dealing with cleaning up, setting up. So when we're looking at microbes, it's not exactly the same, but it's something similar. So I'm looking at microbes through a scope. I'm not looking at their DNA. I don't know what their name is specifically. I don't know exactly what their favorite food is, and I don't know exactly what their function is. But what I do know is that I can see diversity with my eyes. I can see a diversity of fungi. I can see different shapes of bacteria. I can see diversity of nematodes, and I can see protozoa. And I know that when I see this diversity of life present, that I have a more resilient ecosystem, right? So the more diversity of life we have in our soils, the more resilient our plants are going to be to drought, to flooding, to anything that might come along and damage the life in the soil. I'm going to grab my water bottle. We're starting to fill up now, so I think I'm going to move forward. So we have a business called Catalyst BioAmendments. Oh, no, wait, let's start here. Nutrient cycling, most important thing. Look, I know most of you understand this. The soil food web basics are very, very popular, but just in case there's a few people in the crowd that don't understand why it is that we would go to such great lengths to try to make microbes, to use them, to even come up and talk about them today, I want to just go ahead and quickly run through the list. What plants are really good at is photosynthesizing, right? Those leaves are solar panels, and they take in the sun's energy and they literally turn it into sugars, carbohydrates, and foods that they make available for microorganisms. So it's almost this exchange that's happening. The plant is using the sun's energy, it's dropping the food into the soil, and it's calling in the microbes that it wants to work with, the bacteria and the fungi, right? Bacteria and fungi are superheroes of the soil. They can eat rocks, they can eat chemicals. Almost all the toxins that are made by us on the planet can be digested by these microorganisms. They create soil structure. They make it to where the moisture can actually be held in the ground for long periods of time. Casey's gonna go into that in depth later. They're also providing nutrient cycling, like that's how your plants eat. And so this is one of the most important things that we could focus on, is the life in the soil. And it doesn't just go to the plants and the animals, it's also to us, right? We are walking ecosystems. We are almost more microbe than we are people. And so what's going on in our soil is gonna affect our plants. It's gonna affect the nutrient density in the plants. It's gonna affect the terpene profiles. That's probably what you guys are thinking about. It's like one of the first things to change when you have a living soil system is those flavors really come out. And it's the same with food. Everyone here has probably tasted a homegrown tomato versus a tomato from the store. And you know right away that you can taste it that it was homegrown in good, healthy soil. And so the next thing I want to look at is the predators in the system. So the amoeba, the protozoa, the flagellates, they're eating bacteria, and then instantly when they release that as poop, they call it the poop loop, that's a nutrient that's available for your plants to uptake right away. So we need to look at all of the microorganisms in the system. It's not just fungi, it's not just bacteria. You've got to have microbes in there that are eating the fungi and bacteria to actually drive the nutrient cycling. The lag. So we started our career with microbes. We went to school, we studied under Dr. Elaine Ingham, Soil Food Web School. You guys are probably familiar with her. We decided we wanted to be consultants. We were growing cannabis at the time. We had small hand-turned piles that we were verifying with our microscope. We used that compost on our cannabis, and it was insane. It was insane how much easier our life got. Our cost of inputs went down by about 70%. We were growing organic, so very expensive. Our plants were healthier than they'd ever been before. I won't say it was perfect, but man, in 1 year, we were able to really bring our stress levels down to a point where growing was fun. It wasn't, this stressful reality. And what we wanted to do was take that and transfer out and use that information in our large-scale food system. That was our goal at first, was to try to work with food growers. So we got our microscopes. We started looking at all the compost that we had access to, and we really didn't find anything that had robust life in it that you could see under a microscope. Maybe a fungal hyphae every now and again, but no predators at all, not to speak of. So we didn't want to be compost producers. That wasn't our plan. We had to become compost producers to be able to have the material that we needed to do our job. So we were lucky. We were able to get permitting here in Nevada County to make hot compost, almost impossible to do. We are not a green waste facility. Nobody drops off waste to us. We actually pay all of our producers to make sure that the materials that come to us are clean, that there's no latent chemicals in them, or that they've been treated with care. And then we continue that care through the whole process. We use microscopes to make sure all those beasties are present. All the fungi, bacteria, protozoa, nematodes, and microarthropods we want to see in our soil are in this compost. So I would say we were lucky getting started, very, very lucky. And we didn't know we would have to charge for this compost. We were assuming, okay, so at massive scale, they're selling compost for $25, $30, $50 a yard, right? Well, once we started getting at collecting our materials, using our microscope, defining our processes, we realized this is gonna be a really expensive product. And we weren't even sure if the market would bear it. But we kept going, and our community supported us. It was insane. We had a workshop. I think we had 3 or 4 clients, cannabis growers, of course. They used the product. It worked well for them, and they told everybody. And then we set to just trying to make sure that not only do we get the education out there, because you got to know why. What is different about compost? Why is this compost $750, $800, $1,500 in some cases? This biologically focused compost is expensive, and why is it expensive? How do you use it? How do you make the most of it? So that's what we put our focus on, is trying to educate our consumers and the people around us to make sure that they know how to use our product in a way that it's the most economical. The second thing is making sure people know how to do it themselves. Because the best compost that you can make is compost at home. And so the idea wasn't to figure it out, like grab the IP and dominate the market. It's more, how can we figure this out and then share what we've learned with everyone so there's more people making this kind of compost all over the world. So it was a whole process of trying to figure out, how do you bag biology? How do you make it shelf stable? How do you, work with a product that's this moist? And it really, a lot of it came down to just giving it a try and then being really transparent with our clients when something goes wrong. There's definitely been times where we've had to pull our products back or, send people new orders because there's nematodes missing or, there's something microbially wrong with the product. But you can't imagine what it's like to take your work, put it in a bag, ship it to someone, and then know that they're going to put it under a microscope. So our client base are those kind of people that are going to take our product, they're going to look at it, and they're going to call us out if it's not looking good. So I'm happy to say that this is our 5th year in business. We don't get a lot of client complaints. And a lot of our original customers, our clients that started in the beginning, they don't really buy our compost that much anymore because it's a living system. And if you're good at what you do, you don't have to keep inputting to a living system. You learn how to nurture it and it grows itself. And I'd also say that, just this morning, I've only had a few people bring me compost samples. If you have them with you, or you're going home tonight and you want to look at the microbes in your soil, we're going to be doing that in the back corner at our table. And already today, I've looked at a few samples that are full of microbes. This is not normal 5 years ago. So it's very cool to see people are learning how to compost and learning how to farm microbes in a real way. So navigating the micro world, I talked about this a little bit, but who's guiding who? There's really no one out there that has all the answers. You look at Dr. Elaine Ingham or any PhD soil biologist, they are paying attention to the microorganisms and actually learning from them. And so what I want to get to today is just, you can pay somebody to look at your soil, but if you want to be a microbe farmer, if you want to have living soils, investing in a microscope and just taking the time to look around yourself is going to teach you more than any class ever could. It's pretty basic, learning how to identify these microbes, at least, on a basic level. You're going to see a lot today that, if you come over to our table, I bet you'll know what a fungal hyphae looks like after this talk. The limitations of the microscope are what I've already said. We don't necessarily know exactly who we're looking at, right? Like what is the name of this fungi by species? We can't tell that in a microscope. Now, with a protozoa and nematodes, we can, but bacteria and fungi, you got to go DNA for that. So I don't know if any of you guys have ever done a DNA analysis on your compost or on your living soil beds, but wow. It's amazing the amount of data you get back, sometimes thousands of species of fungi to go through. And so this is a typical soil report. When we're taking data, we're working with large-scale farmers. What's great about these tests is it's an estimate of biomass, right? It's not perfect. It's gauged by the human who's running the microscope's eyes, so there's going to be differences between what I see in a microscope and what you see in a microscope. But in general, if the sample's good, you're going to see lower bacterial numbers, you're going to see higher fungal numbers, and you're going to see flagellates and amoeba present. You're going to see nematodes present, and you're going to see less ciliates and oomycetes. But when we look at the data in this way, it's very blanket, and a lot of times it's good versus bad. Like, we don't want to see the bad guys, but we do want to see the good guys. And then, who decided who's a bad guy and, who's a good guy when we have such limited information? I mean, even when we look at DNA and we can say, okay, you're Fusarium, you're a bad guy. Well, not really true. Fusarium's going to perform different depending on its environment. So in an environment where there's not a lot of diversity, there's not a lot of support in the system, Fusarium's going to just wipe out plants. It's going to eat them because that's what it's got to eat. Now, you put it in a different environment where it's got a different group of microorganisms around it, and now it can be working with the plant as an endophyte, actually providing benefit. So some of the worst guys are not always bad. It's just the environment that they're in. Now we get into all my videos. I snuck them in early. Farming for exploration, right? When we're exploring to learn, it gets exciting. And so this video in specific it really opened my mind up to understand that these single-celled microorganisms they are actively operating. They are living in the soil. You can see this test Aerobic, its pseudopods are out, it's moving. In real life, this video probably took me an hour to shoot because they really move that slowly. I sped this up so that I wouldn't bore you guys and hold you here for the whole day. But I want to point out that, you see, it's just getting ready to happen. It's going to grab that aggregate and it's going to pull it inside of itself. And what I like about this video is you almost see it make a decision to move. It's headed out in this direction, and then all of a sudden it goes, wait a minute, there's a snack right there. And it turns and it eats. So these critters, they're interacting, they're hunting. There's an entire ecosystem in one drop of water. When you're looking at this, it's 1/4000th of the slide. The slide is one drop of water. Can you imagine, when you scale that up to one of your pots, how much life is living inside of it? Mind-blowing. And so, the big thing is, when we're trying to understand what we're looking at, we're just observing interactions. We're using the morphology, what the microbe looks like, right? I know this is an amoeba. I can see its pseudopods. I can see its test, which is its shell. It actually builds that shell with those pseudopods, by the way. It reaches out into the solution, it grabs silica, and it literally builds that test, which is incredible. I used to be in natural building. It's not that easy to build this kind of shape. It's difficult. You start to gain an awareness of what their natural ecosystems look like. And if you're working with living soil and you do have a microscope, I recommend getting out into nature. Go to an old growth forest, go to your local forest, check out your bird feeder water. Anything you can think about looking at to observe microbes, do it. Because you start to see, where do they live? What do they do? Why would somebody say that trees enjoy a 500 to 1 fungal to bacterial ratio? Well, if you look at an old growth forest, you'll see how obvious that is. I'm going to show you some slides pretty soon that are covered in fungi. You can't even see bacteria. So this is what I want to show you, is just how easy it is to see in a snapshot the difference. Right? We don't need to know how to account for fungal biomass in order to learn something from the microscope. We don't have to be heavily educated. I am certain that every single person in this crowd can look up here and tell me the differences, right? You can see compaction through the microscope. If I were to put a compactometer on the dirt side, I wouldn't be able to press it into the soil at all. There's no space there for water. There's no space for roots to grow. There's no space for microbes to live. So how useful do you think my compost extract is going to be if I dump it on the soil? It's not going to work very well, right? So using this microscope to tell how frequent are we going to, how often are we going to need to add compost extracts and teas? How much compost do I need to put in? Where am I at in the system? Do I have a thriving ecosystem at all? Well, on the left-hand side, you put your soil or your compost under the microscope and it looks like that. I can almost guarantee that you're not having a lot of problems with your crop. Because in one view, 1/4000th of a drop, we can see 2 different strands of fungal hyphae, plenty of bacteria, and up there is a predatory protozoan. It's a testate amoeba. We can also see some fungal spores right there. There's a lot that we can conclude from just looking at one view. And now imagine if you just keep going. You look at 25 different views, you look at 100 different samples. Before you know it, you start to get a feeling for the microbial world and what you're seeing. Although you always have to remember that you really don't know, so it's a guess. Other things you can see, and I've always been obsessed with the organic material in soil. And you hear this all the time, you want to get your organic material percentage up to at least 3%. 10% is amazing, right? That's the common. And so how do you get organic material in your soil? Do you go to the dump and buy 40 yards of compost and mix it in? That's what a lot of people do. But another way to get organic material into your soil is you're using plants and microbes. Because what is a microbe than organic material? It's a life, and it replicates itself, right? So it's gonna live, it's gonna eat, and it's gonna make more of itself. So it's not just the actual leaves and the sticks and the compost that's the organic material in the soil. It's gonna be the plant's roots that are growing down. It's gonna be all the material that gets dropped off, and then it's gonna be all the living bodies that are in that soil. And so I've got some photos here that just show you some different types of organic material. And when I'm looking through the sample and I see this, that's all food for microbes. And so it's, you can see up there cellulose, leaves, paper, cardboard, wood. When it comes undone and starts to break apart, it looks like these giant springs. The microplant parts, they literally look like leaves, just beautiful replicating cells over and over again, gracefully coming apart as the bacteria eat them. The aggregated mixed organic material, when you start to see the fungi reach out and grab the organic material and pull it together and then web off to another bit of organic material, this is how structure is created. Fungi binding sand, silts, clays, organic materials all together into aggregates to feed itself, basically, but at the same time creating structure, cleaning groundwater, feeding our plants. Incredible. On the other side, that's wild forest soil. That's not old growth soil, but that is just incredibly beautiful fungal structure. Like, that's what we want to see in our soils. And you can imagine that's all organic material. And those are carbon pipes. So this fungi is growing, and through time, it's reinforcing these carbon pipes. It's laying more and more carbon into its own structure. And that's why they say that you're going to be sequestering more carbon when you have high levels of fungi in the soil than when you don't. Because whether this fungi is alive or dead, that pipe work, that stays in the soil unless the soil goes terribly anaerobic and the bacteria take it apart and eat it. It'll stay there for thousands of years. Don't quote me on that, because again, I don't know, it's a guess. But they can stay for a very, very long time. All right, this is interesting too. This is on almost the same property. The top is heavy minerals. This is something we can see in the microscope. We look at a sample, we can say, wow, this is really loaded with minerals. There's not much organic material in it at all. So that top sample, they just sprayed, I think it was glyphosate, and they spray it all the time. They manage all of their plant material by just, glyphosate over and over again. Now, same crop, organic crop, just across the street, they use cover plants. And so you can still see all that heavy mineral. It's almost the same as the first sample, only you can see the aggregated humic and fulvic acids. You can see that fungal hyphae right there. These are the aggregates. And so there's not much different about these two properties except for the fact that there's herbicides used. And there's actually a slide in Casey's presentation where the farmers came over, they just sprayed glyphosate and they wanted us to look at it under the microscope. And you see this crystal, it's like a very sharp crystalline structure, and the bacteria have backed away from it, and there's no life at all, in a huge perfect circle right across, like right around it. And as we went through the entire sample, we saw all these voids of life. And to me, I was like, I don't know for certain, but I was assuming it was the glyphosate. All right, so this is another good example. I want to talk a little bit more about anaerobic and aerobic organisms. There is no war going on between the anaerobes and the aerobes. They are a team. They work together on this planet to keep, our soils healthy, the whole planet healthy. This sample was taken from an old growth forest. It was raining, and we didn't have very much time at all, so we ran through the rain. The ground was quite saturated. It had been, raining for a few days. This is from Corvallis, Oregon. And so, the conditions were quite moist, almost flooded. And so while I was trying to get this video of this really beautiful testate amoeba, you see how spiky it is, how protected it is? I believe that the reason that they create these spikes is because in an old growth forest, there's not much bacteria. And so they don't really have a chance to eat as much as they want to. So I think they've actually protected themselves with these spikes, so it's a little bit easier for them to live. But while I was trying to get the film of these, beautiful testate amoeba, this rotifer comes in. And you can see the rotifer woke up because suddenly there's food. We're in a healthy forest system. There's diversity in our aerobes, and there's diversity in our anaerobes. And so as you work with a microscope, and if you practice and you play around with it a lot, I like to drive my samples anaerobic just to see what happens. And when you see a massive diversity of aerobic life, when you drive that sample anaerobic, you're going to see the same diversity on the other side. So as you grow your aerobes, you grow your anaerobes. It's just the way that the system works. And so when we talk about, when you hear soil food web people talk about anaerobic conditions being bad, it's because of what can happen when there's not a thriving community there to mitigate these issues, right? If you don't have a diversity of anaerobes to wake up when there's too much bacteria, what's going to deal with it, right? And so some of those organisms, if you don't have a really, really nice, diverse ecosystem, some of those organisms are going to produce nasty compounds that aren't so great for plants. But when we have a thriving ecosystem, when we have plenty of bacterial diversity, plenty of bacillus in the system, when it goes anaerobic, there's not going to be these horrible compounds created, or not as soon anyway. So this is why the difference between just straight anaerobic and anaerobic ferments and preparations is that when we're doing preps, we're doing certain things to make sure that we mitigate this anaerobic system, right? There's a little bit of salt used or sugar or, KNF isn't my specialty, but I spend a lot of time talking to people that make these kind of preps and looking at them under the microscope. And so, yeah, I think it's fun to watch everybody kind of living together. I think there's even an extra lag because of my video. All right, so this is, the video's not playing. This is an anaerobic nematode extract. So I don't know who in here is familiar with nematode extracts, but I'll go ahead and describe them really quick just so you understand what I'm talking about. When we take 1 drop of water to look for nematodes, typically we're going to see 1 or 2. It doesn't tell us a lot about our overall system. What we want to see in our soil is all the different trophic levels of nematodes. We want to see bacterial feeders, fungal feeders. We want to see predators. Probably don't want to see root feeders, but 1 or 2 isn't going to be the worst. But a nematode extract is a way to take a handful of your soil or a larger amount of your soil and look at all of the nematode life that's present in it. Now, when soils flood, nematodes dive. That's what they do naturally. They need to get out. They want to get out of town. They don't want to be flooded. They want to go somewhere where there's not so much water. And so the way that we kind of force these nematodes down is we have a funnel with a tube and a clip at the end of it that's filled with water. And the compost is submerged inside that funnel. And the nematodes swim down to the bottom. I cap it, I pull it out, I filter it, and then I observe them. It doesn't kill them. We can actually collect them all. They stay alive for quite a long time. Nematodes are pretty freaking resilient to anaerobic conditions. And that does make sense because they live in the soil. And you can imagine, we just had a rain that was I don't know how much rain we got in 2 weeks, but it was insane. And so to imagine that that's probably been happening through all the history of time and nematodes have, learned how to survive, it's no shock that at 72 hours in an anaerobic environment, there's still nematodes alive and present. What I want to point out is all the organic material that's still around. And I want you to look at that fuzziness. There's a fuzziness around everywhere. Kind of the bacteria have started to come together, and it is what they're getting ready to do is they're eating, and then they're going to go dormant. And so they're covering the surfaces of the organic material and they're breaking it down and then they're reproducing at a rapid rate. And so you're seeing those ciliates woke up in response to that. They're like, oh yes, bacteria, let's have a snack. And so you can see in the video, there's some aerobic organisms around, but it's mostly the ciliates. And we are seeing some diversity of ciliates here. You can see a stellate trichome. Let me see, where is that? Right there. That's a part of a plant that protects it, so things don't want to eat it. You could think comfrey, that spiky material on the leaf would look like this under the microscope. And so what I want to show you is how this anaerobic situation goes over time. Because what I did was I left this sample in a test tube, and I waited for a lot longer, and then I checked it out again. So this is a week later, and you can see there's not much organic material left at this point. There's also not a lot of diversity of ciliates, but there's one there that is really happily reproducing. And I like this video because it almost seems like it wants to get away from itself. It keeps running head first into the aggregates, and I think they're just trying to split away from each other. But that's what they look like when they reproduce. And so it's very interesting to watch these environments collapse, because what goes on is all the organic material just disappears. And where does it go? It gets blown off as gas. And so anaerobic processes are going to be, like, the anaerobes, they want to get rid of the organic material quickly. That's kind of their function. So think about when a deer dies in the woods or an animal dies in the woods, right? There's all these larger predatory creatures or scavenger creatures that are going to eat it, but what would happen if we didn't have a way to just make that disappear, right? So these bacteria come in, they're going to start to decompose in an anaerobic way. All of these protists wake up and eat them, and before you know it, there's no organic material left. And so that's one way that we can lose the organic material in our soils due to flooding, right? We don't have the right diversity of organisms there. Then it floods and sits for, a week, 2 weeks. And those microbes go ham, and they eat a lot of the organic material that's present in the soil. What I want to point out is this is a very famous, right here. And it's not a great video of him, but it's a tardigrade. And the tardigrade is just fine. The nematodes might be gone and dead, but the tardigrade is still surviving. All right, here's another. This is like probably, as far as nematodes go, I think I'm pronouncing it correctly, Criconomoides. This is on this side, this is a root feeder that messes with a lot of our fruit tree crops. They're known for being some of the worst of the worst. And I think not of all the microbes, I'd say this is probably the second worst in people's minds. But let's just think this through. Are root feeders evil? Are they bad? They're grazers. So root feeders are vegetarian creatures. They like to tap into roots and eat them. And the main problem with them, the reason that they're getting, taking over and becoming such an issue in the soil, is we don't have anything to eat them present, right? And so it's just like with deer. If there's predatory creatures, deer aren't the plague of our existence. They're out there eating grass every day. Now, if we don't have cougars, we don't have bobcats, we don't have anything that eats the deer, they can become a huge problem from overgrazing and overpopulating. And so it's a lot like that with our nematodes in the soil. Often when I use the word nematode around farmers, I'll say, we need to get some nematodes, some diverse nematodes in your soil. And they say, no, I've just sprayed nematicides for the past 5 years to make sure not one nematode is growing in my soil. And the truth is by doing that, you're creating a reality where these guys are able to thrive even easier because they're the least sensitive of everyone. And speaking about nematodes, I don't have a good slide in here, but I just kind of want to talk to you about the trophic levels of nematodes. So some of them are going to reproduce, in a week. They reproduce really, really quickly. They have eggs, they give live birth. They're just, bacterial feeders, quick. Some fungal feeders as well can reproduce very, very quickly. But some fungal feeders, they can only reproduce once a month, or maybe twice a year, or maybe only every 3 years. Now, with predatory nematodes, some predatory nematodes take years to get to the age that they can actually reproduce, and then they only have one young per year. They're like people. They can make one nematode at a time. So can you imagine how hard it is to get these guys back into the system after we've wiped them out? And who's thinking about the microbes that are being wiped out, right? These guys are invisible. They're microbes. Micro meaning we can't see them with our eyes. It is very hard to care about something that you cannot see with your eyes. It's hard to protect the lives of something you can't see with your eyes. And it's really funny because I always think about the whales. And whales they're the biggest animals on the planet. But until people started filming them and letting everyone know that they were dying, no one cared about the whales, right? But now everyone cares about whales. Like, whales are coming back. They're coming off endangered species list. You see them everywhere. And I think it's the same with the microbes. Like, this is part of the passion, is making people understand, helping people understand that with a simple tool, we can observe these lives, and that these lives are some of the most important on the planet. They are the foundation of everything. We cannot eat without them. Our plants cannot eat without them. If microbes were to disappear today, it would all collapse. And so I find it very, very important to think about the idea of conserving these organisms. And the best thing about it is we don't have to go out of our way and start a whole campaign doing something different. If we're growing plants, we can be microbe farmers. And by being microbe farmers, in essence, you're protecting the little guys. And yes, we don't know exactly who we're protecting. We don't know if we've saved a very rare species of microorganisms that was on the brink of collapse, but you could be. You know, and until we understand, I think it's just a great thing to focus on. No pun intended. So this is, it's kind of hard to see on the big screen, but these are the one organism that I really want to warn you about, and it's Spirilla and spirochetes. These guys, if you could see them well, they look like a corkscrew spinning through space. So those are all bacteria, super large bacteria. And these guys are pathogenic to people. Not all of them. There's many species of Spirilla and spirochetes, but some of our worst diseases, some of the stuff that really, really is not great for people, can really take human life, are connected to these organisms. And so of all the bad guys, and I won't even put these into that category, these are the ones that you want to make sure you wash your hands when you see them under the microscope. Yes? Yes. So I'm not moving anymore, but all of these, there's thousands of them all through the sample. It is really, really rare to see this kind of domination of Spirilla. This is the only sample I've ever seen. This was from agricultural soil. There was nothing else in it except for Spirilla. I have no idea what caused that condition. But out of the thousands and thousands of samples I've ever looked at, this is the only one that looked like this. So another thing that we can see, another association, interaction with microbes that we can see with the microscope is mycorrhizal colonization on the roots of our plants, and also microbial colonization of the leaves. And so later we're going to get in much deeper into compost teas, compost extracts, all the tools that we use for the work that we do. But with compost teas, you're going to be spraying them onto a leaf surface, and you want to make sure you get good coverage. And so one of the tools that we have to make sure that we're getting good coverage is an epifluorescent microscope. My microscope that I have back here in the corner is an epi microscope. It has an attachment to it. And what it means is I can stain these microorganisms. There's a number of different staining agents that I can use. And then they glow if they're active. And so what you're seeing on this leaf surface, it's not really a leaf, this is a petal, it's an orchid. It's actually that same orchid. You can see all the little red spots. Those green dots are bacteria and fungi. So we were doing a study trying to see how much coverage were we getting, and we were doing pretty good. Now on the roots, the mycorrhizal colonization is going to glow much stronger. And the fungi is actually really amazing to look at because it just looks like it's glowing in the dark. Just like it is when you look at it under the microscope, only very avatar-y. Okay, now we're getting into my favorite part, show and tell. This is a purple testate amoeba. This purple testate amoeba started showing up in my compost 2 years ago. Really, really excited to find it. I didn't know they came in those colors. But what's really cool about this is I was taught at the Soil Food Web School that amoeba only eat bacteria. And so when I found these fungal spores inside my testate amoeba, I found that to be peculiar. And I started reaching out to different people that study these organisms in particular. They spend all their life just studying amoeba. And it turns out that, yeah, as you go through succession, those amoeba that live in the forest, of course, they're not eating bacteria. They're eating fungal spores because it's a fungal-dominated environment. And what I like about this actual amoeba is it's just easy to see it growing in soil. A lot of testate amoeba are clear and they kind of look similar, but this one's big and chunky and purple. So when I put a compost extract out on the soil and then I look at the soil and I see this purple testate amoeba, I know that's from my compost, and I feel pretty good about it. This is another variety of testate amoeba. So they come in all shapes and sizes. This is called Arcella. Arcella they're typically living in forest systems, and I think they're quite beautiful. They're really round, and as time goes on, they get darker and darker and darker. Like I said, they build that shell. So as that amoeba is living in the world, it's investing in its shell, it's adding to its house, it's upgrading. And the mineral that it uses is that color, right? So as it continues to add to its shell, it just gets darker and darker and darker. This one's pretty young. It's still got some yellow to it. Some of them turn almost red. They're super pretty. And they also remind me of just little vases or so. The structure's gorgeous. Sorry, guys, I'm having struggles with I think I'm too far forward now. Nope, I was right where I was supposed to be. So those little critters you just saw that look like candy corn, that's actually from a river. And so I think I've gotten to the point of the presentation where we're totally out of soil and just like wildly checking out whatever I want. This is from a river. It's a river that surrounds a redwood forest. And this is just an incredible diversity of diatoms. Diatoms are algae that live in glass houses, organic glass houses, by the way. That's pretty impressive. They're the only organisms on the planet that can grow glass. And they're often just so beautiful and ornamented in the way they look. That color that you see, that orange and yellow, that means they're photosynthetic just like plants. So they can take sunlight and they can convert that into energy and food. And yeah, they move. They move around a lot. You wouldn't think diatoms move, but they like wiggle and shake. This guy, talk about something I don't know. I have no idea what this thing is. I watched it for about 45 minutes, just, wow, like what the heck? I find when you get into water environments, there's so much diversity of organisms and, so many of these little beasties that you're seeing are actually a form of something else. It's the larval form. And so figuring out exactly what it is isn't easy. Identification it's a struggle. The harder the insect is to capture and get on film, the harder it is to figure out who it is. So this could actually just be maybe a mosquito larva or something like this. I have no idea. But this is the fun stuff about the job, is when you have time to just grab a sample and just, go with it. This is another little beauty. Those little balls that you see bubbling around, those are algae. This guy's getting ready to set off its vortex so that it can force all the organisms around it to fall into its mouth. And you can see inside its tummy there was some unfortunate creatures, a lot of them. I think this is a ciliate, but I really don't know. We don't see a lot of this color and complexity in the soil. Things are a little bit slower there. In the water, everything can free flow and move around so easily that there's just a lot more going on. So this is from a stagnant pond system, right? And we look at it and you can see, if we see these balls, like sometimes with our compost, we use NID water, or we'll use, the water goes from the NID into a pond and then into our compost. So we can actually use these organisms to detect when our pond has too much algae in it. I'll look at a compost sample and be like, where did this algae come from? Well, it's coming through the pipe, through the waters. But this is very typical to what a stagnant pond sample is going to look like, something that's not aerated. And so this is something that Casey brought up earlier. A lot of people will get our compost, they'll look at it under a microscope and they'll go, oh my gosh, nothing's moving in there. Why? And it's because aerobes they don't move very fast. The earth, the planet is typically aerobic, like most soils are aerobic. Anaerobes are waking up when something happens. When there's a flood, when there's, something's going on, something's died, the situation is void of oxygen. And that situation is only going to be around for a limited amount of time. A flood comes in for a day, 2, maybe just a couple hours. So these anaerobes wake up and they just get to action because they only have a short time to eat. So they're moving and they're zooming and they're active and they've got to get their food source now so that they can go to sleep and insist and live for another day. And so your aerobic organisms are basically going to just be sitting there slow. Fungi doesn't move much. So, I guess from all of this, the takeaway is there are ecosystems every single place we look. Your beds are an ecosystem. Your body is an ecosystem. And it's all connected together. Paying attention to the microbes, working from the ground up, from the soil up, is going to reflect health through the whole community. We like to say healthy soil, healthy plants, healthy people. And so, if anything, I know this wasn't the most actionable presentation, but the second one will be, I promise. If anything, I hope that some of you are a little bit more interested in microbes, and that when you leave today, that you invest a little bit more time in figuring out what's going on in your soil and how you can be a good host for these little microbes so they can do their good works. End of show. There was a thank you slide, but all it said was thank you. And it had a big test-date amoeba on it. So thank you guys. I really appreciate your time. Thanks for sitting and listening.